Hello everyone, my name is Hannah Brock, and today I will be giving my presentation on the parasite Toxoplasma gondii. Here we will be going over the taxonomic classification of Toxoplasma gondii. Very briefly, taxonomy is the system of naming and classifying different organisms based on a hierarchy. And for this parasite, its kingdom is Protista, its phylum is Apicomplexa, it is in the Conoidocida class, in order Eucoccydiorida, in the Sarcosistidae family, and its genus is Toxoplasma and species is gondii. So where can this parasite be found? Interestingly enough, Toxoplasma gondii is actually found all over the world. Definitely though, rates of infection are highest in areas that are particularly hot, humid, or at a lower altitude. So anything along like the equator, or something more tropical is definitely going to have higher rates of infection in those areas. And the reason for this is because the oocysts from this parasite actually survive best in these conditions. Now we will be going over the different nutritional requirements for this parasite. T. gondii is an obligate intracellular parasite, which basically means that it absorbs nutrients directly from its host. Its main source of energy is glucose, and that can be found in living cells and in the intestinal tracts of the host that it's in. In the likelihood that glucose is not present or not much of it is around, it can use other carbon sources to generate energy. A couple of examples of this would be glutamine, alanine, and lactate. When looking at Toxoplasma gondii under a microscope, you will notice a few distinct characteristics. It usually sits between 4 to 8 micrometers long and about 2 to 3 micrometers wide. And if you look at the image on the slide, you can see that it has a tapered anterior end with a blunt posterior. T. gondii also generally has a larger nucleus, which you can also see in the image. The darker spot on each one of the little organisms would be that larger nucleus. Now we will be talking about transmission of Toxoplasma gondii into humans. So members of family Felidae are the definitive hosts for this parasite, and members of family Felidae would be any type of cat. In this particular instance though, for this specific parasite, the definitive hosts are usually domestic cats. So unsporulated oocysts are shed in the cat feces, and intermediate hosts become infected after they ingest these oocysts that have been shed. And these oocysts are generally going to be in their water, in their food, anything they will encounter in the environment. And these intermediate hosts are birds and rodents. So once inside of the intermediate hosts, these oocysts will develop into tachyzoites and then tissue cyst bradyzoites in their neural and muscle tissue. The cats will consume these intermediate hosts, and then they will become infected with the bradyzoites, the Toxoplasma gondii bradyzoites. And then humans in turn become infected when they come into these cat feces that have the bradyzoites. And another method of transmission is actually from consuming raw and undercooked meat. So once inside the body, tachyzoites of Toxoplasma gondii undergo active invasion. So they will push through the moving junction of the host cell, and they will enter via the parasitophorous vacuole. This parasitophorous vacuole is a structure that protects the parasites as they develop. And once the tachyzoite enters the host cell, parasite proteins are then secreted from different organelles in the parasite. Here we will be going over the mechanism of host invasion. So the transitory structure is the mobile junction, and that's assembled by Toxoplasma gondii and host cell proteins. And this is going to be beneath the host cell plasma membrane. Rupture proteins and host cell proteins they form a molecular complex that allows the parasite to attach to host cell actin. And the rupture proteins RON2, those are on the host cell cytoplasm. They bind to AMA1, which is secreted by micronemes, a organelle in Toxoplasma gondii. And once inside the parasite, the cytosolic domain of AMA1 will actually bind to the actin myosin motor, which will then allow the parasite to move towards the parasitophorous vacuole and continue to enter the host. Toxoplasma gondii has a couple of important virulence factors that help it infect the host cell. So these would be the ROP16 and ROP18 proteins. And these are serine and threonine kinases. So for the first one, the ROP16 protein, this is going to phosphorylate STAT3 and STAT6 transcription factors, which will then repress the IL-12 signaling. And then from there, IFN gamma cytokine, that won't be able to be produced. And then because of that, the IL-12 and IFN gamma, these are both necessary for the host to survive infection. So if this is repressed and the cytokine can't be produced, then the host will not be able to defend itself against T. gondii. And for the ROP18 protein, this is going to phosphorylate host proteins, which causes acute virulence. When a human accidentally ingests the Toxoplasma gondii parasite, it causes an infection called toxoplasmosis. Most often, when someone gets infected with toxoplasmosis, they don't generally have any symptoms. However, if symptoms are present, they're generally going to present as flu-like. So you're going to be getting fever, swollen lymph nodes, rash, headache, muscle aches, everything along those lines. However, immunocompromised people, when they get infected with toxoplasmosis, they generally will have symptoms. And these are going to be similar, and they will be shared with the symptoms from healthier people, but they will also have a few other things and be a little bit more severe. Examples of these would be breathing problems, fever as well, cough, confusion, poor coordination, and sometimes even seizures. So what specific population is generally at risk for toxoplasmosis? As mentioned before in the previous slide, immunocompromised individuals would be most susceptible to getting this infection. And so basically anyone who has HIV, AIDS, is receiving any sort of cancer treatment like chemotherapy, or anyone who received an organ transplant, they would be very high risk for this infection. Also, infants, if their mothers were infected during or just before pregnancy, they are also at very high risk for contracting this infection. So under what circumstances could you get this infection? When you're eating undercooked meat, there's always that risk, or coming into contact with cat feces or anywhere cat feces could be present. So like in the soil, just outside in general, stuff like that. And also if you live in a particularly hot or humid place, then you're also at a higher risk. However, you can reduce the risk of contracting toxoplasmosis. Just be very thorough with washing and peeling fruits and veggies, cooking meats completely through to the optimal temperature, and just proper hygiene, sanitation, and wearing gloves and protecting yourself when you are cleaning out litter boxes, or if you're outside anywhere a cat could have been. If you believe that you may have toxoplasmosis, then you can do a series of tests to confirm that. So typically, serologic testing is the most common way to test if you have toxoplasmosis, and this basically just measures your immunoglobulin G levels in your body. And if it is necessary to estimate around when you were infected, there's another test that you can do to measure immunoglobulin M levels in your body, and that will give you a general idea of when you could have contracted this. There are a few other methods that are used to just test for toxoplasmosis, but they aren't used as frequently. A couple examples of these would be staining tissue samples, testing cerebrospinal fluid, and isolating T. gondii from different bodily fluids like blood and saliva and whatnot. So if the serologic testing comes back positive for toxoplasmosis, T. gondii actually has a few drugs that it's susceptible, so there is treatment for it. The first one would be pyrimethamine. This is the most common drug that is prescribed to toxoplasmosis patients. What this does is that it fights infections caused by microscopic organisms in general by blocking the body's use of folic acid. Sulfadiazine is another drug that is prescribed as well. It is very commonly prescribed with pyrimethamine, and this antibiotic just works very well for that. And the third most common one would be leucovorin calcium. This is also commonly paired with pyrimethamine, and it helps to fix the effects of that first drug on the body's folic acid activity, because pyrimethamine, it like I said, blocks the body's use, and so leucovorin calcium, that is brought in to restore that. So Toxoplasma gondii actually has pretty low genetic diversity, and as I mentioned before in previous slides, it is found worldwide on all 7 continents, so drug-resistant species are found worldwide as well. And a few examples of drugs that it is resistant to, these antibiotics, clindamycin, spiromycin, and azithromycin, these are all antibiotics that it is resistant to, and resistance for this is encoded in the rRNA of the parasite. And before, I did mention that Toxoplasma gondii is most susceptible to pyrimethamine and sulfadiazine, or at least these are the 2 most commonly prescribed drugs to fight toxoplasmosis. However, in some instances, there are certain strains of T. gondii that are resistant to these 2. So for pyrimethamine resistance, analogous amino acid substitutions in the Toxoplasma enzyme are going to be responsible for that. And studies have shown that there is a recent rise in sulfadiazine resistance, but that hasn't been proven as of yet. Resistance to pyrimethamine in Toxoplasma gondii is going to use in vitro mutagenesis. So T. gondii achieves this resistance through a series of single point mutations, specifically in the DHFRTS gene on their genome. Specifically, the T83N mutation in this gene, that is going to be what's responsible for T. gondii's resistance to pyrimethamine. Interestingly enough, though, if there is another single point mutation in the S36R mutation, if that's paired with the T83N, that increases the resistance to pyrimethamine. And if it's S36R and the F245S mutation, both of those paired with the 283N mutation, that is also going to greatly increase the rate of resistance. And that is going to conclude my presentation on Toxoplasma gondii. The rest of this here I have is just a series of the works that I cited for this presentation. And I just want to say thank you for listening and have a good day.